Hi friends, welcome to Brian Academy. In this video, I'm going to talk about enzymology. If this is your first time coming across our video or visiting our channel, don't forget to like, share, and subscribe. It's very, very important. Now, what I'm going to be focusing on as far as enzymology is concerned, I'm going to talk about the first point. I'm going to talk about the definition of enzyme and enzymology. Then I'm going to focus on the nature of enzyme itself. Then after the nature of enzyme, I'm going to describe the structure and uh, the major part of an enzyme that helps to catalyze the reaction. Then the next thing I'm going to focus on is actually to talk on about the specific properties, or let me just say general properties of what? Of enzyme. Then after that, I'm going to talk about how enzyme work. How enzyme work. So these are the four major points I'm going to be focusing on in this video. So let's start. Now, the first thing is definition. Starting from enzymology. Enzymology is basically an aspect of biochemistry that has to do with the study of enzyme, right? It has to do with the study of what? Of enzyme. Then, as far as enzyme is concerned, what are we going to study when it comes to enzyme? We are going to study the structure of an enzyme, the function of an enzyme, and how enzyme perform its works, right? How it helps to catalyze what? A reaction. Then, what is this enzyme? Enzymes are biocatalysts. I know you guys you've actually heard about catalyst before. Catalyst, right? Actually heard about catalyst before in chemistry. And catalysts are basically substances that help to speed up the rate of chemical reaction, right? Now, let's take it and relate it to the biological system. Let's say, okay, we are talking about catalysts as far as the living system is concerned. The enzymes are basically catalysts. They are biocatalysts because they act within the living system. They are organic catalysts, or you simply say they are biocatalysts that helps to speed up the rate of biochemical reaction, right? The reaction that occur in the body are actually termed as what? As biochemical reaction. But the enzyme, or let me say the biocatalyst that help to enhance the rate of this biochemical reaction, they are referred to as what? As enzyme. And one thing about enzyme is that enzymes are usually proteinous in nature. That is, they are what? They are protein, right? That is, their nature is basically what? Protein. And they help to enhance the rate of what? Of biochemical reaction. That's basically what enzymes are. They are biocatalysts, or simply they are bioorganic catalysts. And that's just it about the definition. Then let's just basically move to the nature of enzyme. And as I said earlier, enzymes are basically, they are usually proteinous what in nature. They are usually protein. Enzymes are what? Are protein, except some for some ribozyme, right? They have catalytic activity, but their nature is not protein, right? So that's just it. Then let's just come to the nature of an enzyme. Let's say I'm having an enzyme, right? And this is going to be my enzyme here, right? And as I said earlier, enzymes are basically what? They are protein in nature. That is, their structure is made up of what? Of protein. And we know that protein are what? Are biomolecules that are made up of what? Of amino acid that are joined together by what? By peptide bond, right? Now let's come back here. Now, if this is basically an enzyme, right? There's a specific part of an enzyme that helps to participate in in a biochemical reaction. This particular part of that enzyme is referred to as what? As the active site. Active site, right? Active site. So this active site is divided into two. Then the first one, let me just say I'm having active site here, right? This active site, right? Is divided into two. We have the binding site. The binding site, and at the same time we have what? We have the catalytic site. Now, I know you guys will want to ask me that why do I divide them into two? That's because these two parts are the one that comprises of the active site, right? The binding site is a special site on the enzyme, or let me just say special region on enzyme that helps to bind a molecule. And this molecule, we call it what? Substrate. Substrate are basically what? They are molecules that enzyme act on to be converted to products. Are you with me? Now, this binding site is actually where the substrate is going to bind to. Then what is going to bring about the catalytic changes in the substrate is first as what? The catalytic site. Binding site what will bind the substrate, while catalytic site will bring about the reaction. It's one that will basically bring about changes that will occur to substrate for it to be converted to products. So generally, enzymes are basically what proteinous in nature, and they are made up of what? Of active site. The region of an enzyme that participates in a biochemical reaction is referred to as what? Active site. Are you with me now? So this is where the substrate will bind to, and this is where all the changes that is going to happen to the substrate is going to what? Is going to occur for it to be converted to what? Product. Now let me just take you guys through this particular diagram you are seeing here. This specific bad boy here that you are seeing here is actually called what? It's actually referred to as what? As the enzyme. This is the enzyme, right? This bad guy that you are seeing here, I'm going to call it what? Substrate, right? And then what you notice is that this substrate actually binds to this enzyme at this particular site. This particular site is actually what I call the what? The active site, right? This is the active site. Are you with me? Then from there, substrate is going to bind to this active site to form what? Something that we call a complex, which is basically enzyme substrate complex. This is basically ES complex, enzyme substrate complex. Now, if you calm down and look at this very well, when the reaction proceeds further, this guy later detach to form what? This particular molecule you're seeing here, and this guy here is first as the product, right? And this guy is still the what? Is still the enzyme. Are you with me? So this will lead us to what? To the general properties of what? Of enzyme. I'm going to bring out the general properties from all these diagrams we are seeing here, right? And the first properties that I would like to talk about, as far as the nature of what enzymes is concerned, is that enzyme, they are what? They are, let me just simply say, let me just move to the side here. Properties of what? Of enzyme. The first thing I need to know is that enzyme are what? Are colloidal, right? They are colloidal. Then the second one that I need to know is what? Is thermolabile. Now, this thermolabile is basically explain the fact that enzymes are what? Are heat sensitive. What do I mean by heat sensitive? Whenever there is alteration in what? In the temperature at which an enzyme perform its optimum function, then the enzyme is either it will become what? Inactive, or it get the what? Denatured, right? So if the temperature is what? Is too high, above the optimum temperature for an enzyme, the enzyme is going to what? Is going to get denatured. But if the temperature is too low, the enzyme will remain inactive. It's not going to perform its function. So enzyme are what? Are thermolabile. That is, they are heat sensitive. That's another thing I want you guys to know. Then another thing I want you guys to know is that enzymes are highly specific. So in this case, I'm going to call it what? Specificity. They are highly specific. Although I'm going to make a separate video on enzyme specificity, because we have a lot of specificity as far as enzymology is concerned. We have substrate specificity, we have group specificity, we have bond specificity, we have a lot of specificity. I'm going to explain it in a separate video. As far as this specificity is concerned, the one I'm going to talk about is that enzymes are highly specific to the substrate they catalyze. That is, to the kind of substrate what they bind to. They are highly specific substrate. Take for example, let's say you take in protein, right? The types of enzyme that's going to act on protein, break them down into amino acid, is different from the one that's going to act on carbohydrates, right? For carbohydrates, we refer them to as amylase, right? For protein, we call them what? Peptides, right? So we have different enzymes. So different enzymes are actually specific for the type of substrate they catalyze. Are you with me now? So in this case, I'm going to talking about what? Substrate specificity. And the reason why enzymes are highly specific to the substrate they catalyze is because the active site of the enzyme is complementary to the what? To the substrate. Now, if you look at this now, this active site and the nature or the structure of this substrate, they are actually what? Complementary to each other in such a way that the substrate can fit into the what? Into the active site, such as a key can fit into what? Into lock. So that is basically models of enzyme action. And I'm going to explain that one in a separate video. So enzymes are highly specific to the substrate they catalyze, right? Then from there, if you look at this, once the enzyme binds to the substrate, it forms enzyme substrate complex, which further dissociate into enzyme and product. That one will lead us to another property of enzymes, which is basically enzymes are not always used up at the end of the reaction, right? So in that case, once enzyme catalyze the conversion of substrate to product, that same enzyme will remain chemically unchanged. In that scenario, it can go back again and pick up another substrate, convert to product. So a single enzyme can actually act on thousands of substrates. right? So that one will what lead us what to efficiency. Efficiency. Enzymes are highly efficient. A single enzyme can catalyze millions of substrates and convert them to what to products, right? So that's basically what we have here. So another one that I'm going to talk about is sensitivity. Sensitivity. In this scenario, as far as sensitivity is concerned, enzymes are highly sensitive to to environmental condition, such as I've explained the first one, which is basically which is temperature. Then the second one that I'm going to talk about, the pH. The third one, we have substrate concentration, we have product concentration. At the same time, we have what? We have inhibitors as well, right? But in this case, I'm going to be limiting everything I'm going to say to temperature and also pH, because in my next video as well, I'm going to explain various factors that can actually affect enzyme action, right? So let's come back here. Let's talk about the pH and also the temperature. I said something earlier. I said there is a particular temperature at which an enzyme can catalyze a reaction. That particular temperature is referred to as optimum temperature. Any variation, either decrease or increase in this temperature, it will affect the catalytic activity of that enzyme. The same thing is what is applicable to pH. Since enzymes are protein, definitely pH can actually change the structure of an enzyme. pH can affect the structure of an enzyme, in the sense that enzymes as well have the optimum pH at which they perform their reaction. Take for example, let's say we have amylase, right? The function of amylase, salivary amylase, actually starts from the mouth, right? And we said salivary amylase require alkaline to perform its optimum function. Then immediately it enters the stomach. The pH of the stomach is highly acidic. This makes this enzyme inactive. In that case, we basically say digestion of carbohydrates does not take place in the stomach, and that is because of the pH of the stomach, which is high. So enzyme, they have their own particular pH at which they perform their function. So this pH is referred to as optimum pH. So they are highly sensitive to this pH or temperature. That's basically what is happening there. So that about the sensitivity. So finally, I've actually covered the definition of an enzyme and enzymology itself, the nature of enzyme, and the parts of enzyme. Now the next thing I'm going to talk about is how enzyme work. Let's move to how enzyme work. Now look at this graph that I have here. This side is basically the reaction coordinates. This is the reaction coordinates, and then here I'm going to represent this as let me simply call it energy of reaction, or you basically call it potential energy. Potential energy, right? Now look at this very well. This is my enzyme and this is my substrate, and at this point I'm having enzyme substrate complex. This is basically ES complex, right? Then from here I'm having enzyme and product. And don't forget what I actually explained earlier, that is enzyme we pick up the substrate, right, to form enzyme substrate complex, which is this guy that you're seeing. Then from there, the enzyme substrate complex is going to dissociate further into enzyme and release the product. So the same thing is basically what we are having here. Then the question is, how does an enzyme act? That is, how does an enzyme act? How does an enzyme perform its function? There's something that we always call energy of activation, or you basically call it activation energy. And this point to this point, I'm going to represent as activation energy. So let me just simply call it activation energy. And what is this activation energy? Activation energy is basically the minimum amount of energy that is required for a reaction to occur. That's in order for enzyme to act on substrate and convert to products, there is minimum amount of energy that the enzyme must meet up, or that the reaction must meet up. That minimum of energy is referred to as activation energy. Now look at this. Imagine we do not have an enzyme. Let's say we don't have an enzyme. Then how can this substrate convert to products? It's not possible, right? How can the substrate itself convert to product? It's not possible. Even though if you carry out the reaction outside of the living system, right, using temperature or and at a normal condition, it will take a very long time for our substrate to convert to product, or no reaction will even occur. So in that case, introducing an enzyme make it easier for a product to form. right? In that case, enzyme basically helps what to lower the activation energy. Then look at this. It's not easy for the substrate to climb this mountain, right? Instead, enzyme will basically what? Will basically lowers what? The activation energy. It lowers what? The activation energy by binding towards what? To the substrate. Right? I'm still going to explain how enzyme lowers what? The activation energy. But basically, an enzyme acts by lowering the activation energy. And what is activation energy? Activation energy what? Is the minimum amount of what? Of energy that is required for what? For a reaction to, what? To occur. Now let's picture it. Let's picture it in this what? In this way. Let's say we have what? A mountain here, right? This is a mountain here, right? And then this is what? This is the substrate. This is the substrate, right? In order for this guy here, this bad boy here, to actually climb this particular mountain and come to the other side, this guy actually need to have what? A potential energy, right? That is either equal or greater than what? That the energy that is required to climb this mountain. That is, let me come back again. This guy needs to process a potential energy that is either equal or greater than what? Than the energy that is required for this guy to climb the mountain. If the energy is not equal or greater, then this guy won't be able to climb this mountain. Imagine this guy, he has not feed, he has not consumed food. So in that scenario, he doesn't have strength to climb the mountain. Will this guy climb the mountain? It's not possible. So this guy needs to possess a certain level of energy for you to be able to climb this mountain. So in that case, the certain level of energy that this guy actually needed is referred to as activation energy. And in that case, enzyme come and lowers what? The activation energy. This guy does not have energy, right? And this guy needs to climb the mountain. Then where this guy is going to get the energy from is basically what? From the enzyme. So in that case, enzyme will basically what? Lowers what? Activation energy in such a way that this particular guy can actually climb the mountain and get what? To the other side. So what I'm basically saying is that enzyme will act on substrate by lowering what? The activation energy. Substrate will convert to what? To the product. Naturally, or let me just say on a normal condition, substrate cannot but convert to product without the action of enzyme. So enzyme acts by bringing the molecules of substrate together. In that case, that is bringing the molecules of substrate together, the activation energy is lowered. So whenever enzyme binds to substrate, the activation energy will reduce. So once the activation energy was reduced, then the substrate will be converted to product. That's basically what is happening. So from this point to this point is the activation energy of uncatalyzed reaction. Uncatalyzed reaction, that is when the enzyme is not present. The normal pathway that the reaction needs to follow is basically this route that you are seeing here. Then once the enzyme is involved, this guy that you are seeing here is basically what? Activation energy of what? Of a catalyzed reaction. So an enzyme acts by lowering the activation energy. In that case, once it's lowered the activation energy, then the substrate will be able to convert to product. So at the end of the day, the enzyme is going to release the product and remain unchanged, so that it can go back to the reaction and pick up another substrate, then convert to product. Right? That's basically the reason why an enzyme is highly specific to the substrate that they act on. Right? And at the same time, that's the main reason why an enzyme can catalyze, a single enzyme can catalyze thousands of substrates. Why? Because they remain chemically unchanged, right? And they will be available to catalyze the same substrate and convert to products. So that's basically everything on how enzyme work. So the point that you need to note here is that an enzyme acts by lowering the activation energy, and the activation energy is the minimum amount of energy that is required for a reaction to occur. So by lowering the activation energy, the substrate is able to convert to product. So that is basically how an enzyme works, right? So let me just quickly come back to this binding site and catalytic site. Now, I said something earlier. I said the active site of an enzyme is made up of the binding site and what? Catalytic site. Now, one thing that you need to take note of is that whenever an enzyme uses this special site, which is the binding site, to bind to a substrate, at that point, right, there's something that we call transition state, right? Transition state is basically what? The state that exists between what? Between the low energy level and high energy level. Right? So at the middle part, we have something we call what? The transition state. Right? So transition state is basically a state whereby a substrate converts to the product, and it is very, very fast. So you can just take this as example. The binding site of an enzyme is actually a special site where the substrate is going to bind to. So once the substrate binds to that particular site, the activation energy has been lowered, right? Because for the substrate to convert to product, it needs the catalytic site. Catalytic site is basically the one that bring about the transformation of substrate to product. So once the substrate is not that, like, it doesn't bind to this binding site, there won't be any change that's going to occur, right? So we can basically say once the substrate binds to the binding site, the activation energy has been lowered, then transition state will be followed immediately because the substrate will be converted to the product, right? So that's just everything about this video. In my next video, I'm going to explain the mechanism better, right? I'm going to explain the mechanism of enzyme action, how enzyme binds to substrate to form enzyme substrate complex, and how enzyme substrate complex decisions into enzyme and also product. So if you enjoyed this video, don't forget to like, share, and subscribe. And also, if you are actually watching from us from TikTok, so make sure you go and visit our YouTube channel, because we have a lot of video there that actually talks about enzymology. So that's just it. Thanks for watching.